Nope, there's another one there on the right.
Yep. Maybe it's their defense pose.
Maybe they just know something we don't. They're not, at least they're not all looking in the same direction. That might be a little weird.
So Science, when we're done with this transect, do we want to stop and look around or continue on to BF?
We'll slowly continue to BF, maybe take us a shorter ship step so we can potentially stop if we see more of those snails we'd like to do a slurp of.
Roger.
There's a different star.
Yeah, our first bigger star. I've seen lots of these little spheroids.
So we have 5 meters left for Herc to finish his transect, and the ship has stopped, so I'm going to put in another short move.
Perfect.
We'll overshoot the 200 meter range just to make sure we made up for the pause in the transect.
Awesome, thanks. Almost to waypoint BF, which is the second waypoint that we've plopped here on the map to help guide us through this deeper part of the terrain that we're hoping to cross. It's probably going to get a lot steeper after BF on the way to BG. Well, actually, maybe that's not true. After BG on the way to BG1, we'll see a significant difference, but we'll probably cross a smaller portion of really steep habitat on the way to BG. And BF is over an area that the backscatter is telling us has a pretty high strong return, which indicates hard bottom somewhere, but it could be very well that it's covered in a layer of sediment here.
Transect complete.
All right.
Wonderful.
So pilots are still moving 036. Now we can stop and zoom if we need to, or stop and sample if we need to.
Excellent.
What more could we ask for?
What a day.
I'm having myself a time.
I guess we could ask for some
get in front of you a little bit.
more interesting features, but
what's our next bearing?
It'll actually be more or less in the same direction. So
cool.
Yeah, we've got about 60 meters to our next waypoint, and then BG is, zoom out here. BG is going to look something more like 046, so still northeast.
All right, Al, let's zoom in.
Hello, thorny head. Kind of has his dorsal fin down, so you can't see it super well.
I know it needs to flare them out. Come on.
Sometimes they do that for us. It's very nice of them.
Yes. Now you can make out the lateral line there. You see that series of pores running back?
Mm-hmm. Kind of goes up and over his gill cover and then back down his side on either side. And it's so amazing to me how rockfish really don't care about this ROV. So many different fish species will spook or swim away.
From this angle, those eyes look incredible.
Yeah, the big dome.
Mm-hmm.
Yeah. All right,
look at the way it's mm-hmm.
Let's come wide, Al. Do have to.
There's this third one. Okay, look back, count the spines, and then the third one should be longer.
Yeah.
Mm-hmm.
It's an interesting gelatinous.
Oh, what is that gelatinous thing? No, it's okay.
We're not gonna be able to.
It's just, yeah, it's fine. If we need to catch up with the ship, that's fine. I don't know what lots of things are down here, so that's quite all right.
In science, are we looking to get to BF and BG expediently, or are you looking to zoom on anything on the way?
Oh, that's a nice hole. I think maybe once we collect these snails, we could move more quickly towards those waypoints. And however fast you feel is safe given the amount of slope that we're encountering too. So I'll be keeping an eye out for the snails, and then once we're finished with that, we can hightail it out of here.
Sounds good. And yeah, we can stop anytime. So
okay. I'm keeping my eyes out for some big snails.
Yep.
Oh, Julie, are you in the science chat now?
No, but I'll work on it. Maybe they were working on reauthorizing my credentials.
Oh, they probably tried it for you then, because it says Julie Bursick joined the science chat, so I think they're working that out for you.
Oh, good. I feel like I'm part of the team. I've been feeling kind of left out.
Oh, we'll get you that chat login soon. I think they must be testing it now.
I ended up
a snail, maybe two right here. Can we do a zoom if we have time?
Oh, what was that?
There was
we were looking for some snails.
Were they back on the bottom of the screen now, or still on screen?
Yeah, they were at the bottom of the screen. I'll refresh my feed to see if I can reduce the lag time a bit.
I think they might be right there.
And it might just be one big one too. It may not be multiple. Yeah, that little blob.
Those two?
I think they're two. Are they two? One and one?
Yeah.
It's hard to say. It could just be a clod of dirt. Oh, I'm saying two.
Nope, I'm still out. They're testing me.
Okay.
It's okay.
All right, Al, let's, I know there's a lot in the water, but let's zoom in a bit.
It's a snail.
That's what we're looking for?
There it is. Yep, I believe so. I'll wait for Daniel's confirmation, but I think we're going to slurp this guy.
And was that a second one up and to the right? Let's zoom out a bit.
Like a clod of...
Oh, that's just a dirt cloud. All right.
Yeah. Yeah, Bathybembix.
It's a Bathybembix?
Bembix. Bathy, and then B-E-M-B-I-X.
And we want to slurp on this one.
Yes, please.
Chambers, this one?
And is it a predatory snail?
Oh, we need to flush. You can go ahead
Yeah.
and do that one. Oh, wait. You might be in the dirt with the slurp. Need to look at it.
Yeah, and
Let's come on.
you have some sediment in there from that first sample too.
Yeah, so we definitely want to flush, but we want to... Is that going to just suck up more dirt?
Well, I think it might be far enough off. You need to zoom in some.
Let's zoom in now.
I could
That's
start
pretty
it.
close.
See?
Daniel's saying this is a deposit feeder.
Yeah, it's sucking
A deposit
stuff.
feeder. Okay. That makes sense. I just didn't know if it was, it's like some of the whelks that go around and
Need to
hunt.
pick it up.
Who's going?
You can do it.
All right.
Daniel's saying that this particular snail has a broad radula with many teeth per row. Rhipidoglossate radula versus whelk that has 5 teeth per row, which is stronger and can cut into meat since it's a predatory snail.
Right. Well, that's interesting. I know radula are like a ribbon of teeth on different mollusks.
Which is characteristic of the mollusk phylum.
Yes. Well, and it kind of sounds like a little bit like maybe like an abalone radula that's sort of taking the diatoms and the microalgal film off of rocks. It's not going to have necessarily a teeth-like radula. It's sort of scraping things.
Right. Yeah. So they have these radulas, but they can all be they're kind of diversified. They've evolved in slightly different ways to suit that niche that they fill. And Daniel has pointed out that those cone snails, those really dangerous ones, have 2 teeth per row on its radula,
Are we good on the
and they're
flush, or do we want to flush here?
modified for toxin injection.
Do a flush. Here we go. Flushing.
That's very interesting.
Mm-hmm.
All right.
He's also saying that abalone have a similar
What jar are we going in?
You can go number 1.
radula as
haven't
this
used any
snail
yet.
here.
That's what, yeah, I would.
Very similar group.
Similar group. Very cool.
Wait.
Oh, it's actually bigger than I anticipated.
Ready?
Is that going to be too big?
Oh, looks like it went in. Let's see.
So the nozzle is smaller diameter than the hose, so if it makes it past the nozzle, it should be okay in the hose. Theory.
So this is in sample jar 1.
Mm-hmm.
Not yet, but will be.
Yeah.
How do you spell the family of that snail? I'm putting it in.
Yeah, Bathy.
Pull the hose out too.
BATHY and then BEMBIX.
You can go in and out.
Oh boy.
Is that it? There it is. In the hole.
Is
lost. There it is.
And another little snail. You got a twofer.
Is that a snail in there?
Yeah,
Yeah,
you got two.
it's like
The big one and the little one.
a family.
Steve, this is 100.
Right.
Thanks.
100.
Alrighty.
Thanks, Lizzie. I was trying to remember what had to go there.
Yeah, no worries. And then I think if we center
Am
the
I good
camera
on the
again and we
arm? I
look
can't.
Yeah, that's fine.
up and left, we might see another one because I think our goal is for 3 of these.
Roger, up and left when we get settled.
Yeah, whenever it's ready. And we can put all 3 in the same sample jar.
All right, let's do the junk zoom. Am I squared up? Yeah.
Good.
I thought I had seen one. Maybe
Yeah.
maybe not.
I still need to
Another.
come over a bit, right?
Seeing any others
Oh,
nearby?
right there in the very upper left. Yeah.
Upper left?
Yeah, so now if we just turn left, it should be center screen.
Oh, yeah,
Oh,
there it
yeah,
is.
yeah.
Found you.
Number 2.
Can't hide
Taking
from
that
us.
one too?
Not in this.
Yeah, we're looking for 3.
To go in the same jar?
Yeah. Thanks. Can't hide from us unless you're an orange cap coral. Sorry, friend.
Look at that trail.
Yeah. Wonder if they were friends. They had a rendezvous.
All right, let's come wide.
Guess that's lined up.
Oh, cool. So Laura Francis is going to join us on VLink soon. She's going to be moderating some public questions. And she's saying, although she can't get the COVID message to go away, saying that the messages aren't on, she's already getting some in the chat box. So
Yeah, if
she'll be
she
joining us.
flips that availability button over and then hits the save button, that message will go away and it will show a green light and that she's available to take questions.
All right, we can zoom in now.
Hi there, this is Laura. Thanks so much, Sam.
All right.
I've done the save settings and it's turned green on my screen. I'm just not seeing it turn green when I log in from the public screen.
Oh yeah, don't worry about it. It takes a little bit for the cache to clear sometimes.
Awesome.
So if you've done it and it looks good, should be good to go. Welcome back.
2.
Great.
Got it.
Very nice. We're
Just in
currently
time.
collecting some
Yay.
snails. Slurping them up.
Slurping the Bathybembex. Verdi
Mhm.
eye?
Yep.
I'll just leave it.
Oh, yeah.
Okay, lucky number 3.
They're like, what is going on?
I know.
Closing their
They
door.
swirled around.
Shut that operculum.
Yeah.
I have a question here if you're available to answer. It's about, can brittle stars be used as an indicator of sediment quality?
They might be able to in terms of if we sampled brittle stars, maybe we could test them for contaminants. But in terms of sediment quality,
That one?
sediment quality with respect to what necessarily, I'm not sure this person might be referring to, but
Well, I know for urchins, isn't it,
Yeah, let's zoom in.
habitat
like halfway.
quality could have better
Nope.
uni or gonad material. So in years when kelp is plentiful and they're getting the right nutritional needs, they tend to be healthier. And I don't know of any ophiuroid studies that are going on, but I wonder if you could sort of
That's a
use it
fish.
as an indicator of health in that way.
All right, let's come on
Yeah,
again.
Well, there's that third spine on the thorn back. I see it.
Oh man, one more
okay, now
snail.
the questions are rolling in. So I guess some of the outreach we've done has been
yeah, but I wonder if
has
this
been good.
viewer is also asking, like, maybe just by the species that we're seeing without necessarily sampling them, if we could tell anything about the sediment that they're in based on just who's there.
That's true.
But I'm not sure. That's a great question.
Oh, how about that?
Yeah.
I
Top right. Easy.
Is that white one? Bob,
Let's zoom in now.
maybe not.
What? Ah, boiled.
No.
It was a snail
There's
until
a-
you said easy.
Oh,
I
that
know.
would be an
Then it transformed. All right, let's come wide.
It almost looks like a shell that been there for a while.
It's a house for sale. Some
There you go.
crab.
There's another question here related to the sampling, which is asking about, are you collecting samples for a particular research goal or just general sampling?
Oh, there's a trail. Does it lead to a snail?
Oh, follow that trail.
Snail
Oh,
trail.
look at
Snail
that.
trail. Oh,
Left
which
or
way is it
which way
going
is
though?
it going though?
Detective work happening. That's a long snail trail.
Dust. Dust
He's gonna be a hardy one.
Oh, I think there
It's
it
leading
is.
to the
Yeah.
it's leading to the mother lode.
Nicely done, team.
Oh no.
Well,
Is it well, that's clearly not a snail. But I think what we thought was at the end of that bioturbation is also not a snail.
let's zoom in now.
Oh, what could it be?
Oh,
Not
maybe it is.
I
Oh,
know, I don't know,
is
it
it a
kinda
different
has
snail? Daniel's
man.
saying third one. Let's wait for the dust to settle. And to answer that question,
Let's
Laura,
come
yeah,
on.
we are collecting these for a specific researcher
weed it out.
with specific goals. So pretty much all of our objectives are pretty specific. We're collecting identified as priority or critical.
Lizzie, I think you cut out, or did I cut out?
Oh, Lizzie cut out.
So with
Oh,
that,
okay.
was that we picked up mud from our last two samples?
We're moving. Yeah.
She had gotten very loud again. I think it was my
Oh.
proximity, so I lowered her level and whammo. I can't touch her volume.
Don't touch Lizzie's volume
You need-
anymore.
oh, it's cutting.
Blowing up my meters here. Oh boy.
Wow, it's
Yeah, I think
this
you'd
could be a while.
better to go on.
Alrighty.
All right.
All this mud
We just went
dabbing,
to zero
picks up a
visibility.
lot of-
And
Yeah. Let's come
we're also
wide out.
taking water samples for environmental
Lizzie,
DNA.
Lizzie,
you
for
cut
a
out
specific
for a little bit
researcher
there.
who's looking at sampling eDNA as a way to see who's around in the water that we may not be
Was getting
seeing
weird.
with our eyes. Okay, my science chat.
Is there another one right up there in front of that thorny?
Front of the thorny head.
Need to get out of this area.
Yes.
It's all stirred up.
Yeah. Which way are we going?
Still 036.
There's something white just above the laser
Is that it right
pointers.
there? No, that's
Down
the
there?
head. We need to get out of
to
this
the-
particular
Yeah, yeah.
like zone.
Okay.
Do we want to take a
We're
step?
just gonna be. No, he could just
Oh,
somewhere
okay.
else. Not right here.
There's a question
Preferably
here
in the
for
current.
the pilots when you get a chance on. It's interesting that you never seem to get tangled in your umbilical. Is it buoyant or does it trail behind you?
If you look in the Argus view right now, you can see that there's
Channel 2.
Yeah, and you can see the tether there, and it has 2 football floats. You can
How
see
about
the
that one
big
up there?
lumps in the tether, and there's also a weight. So there's a weight suspended on the tether below Argus, which pulls it down, and then the 2 football floats are designed so that you get kind of an S bend in the tether, and that sort of takes up the slack. And the idea is to keep it from, getting kinks in it like a garden hose.
All right, Al, let's zoom in.
We've been trying to get an image of the slick heads that we've been seeing, and I think this might be a larger one of them.
Another shell.
That's just a shell, right?
Oh.
And there
Yeah.
he goes.
All right, let's come on.
It's okay. And we were able to see that this was just an empty shell, so double dip. There's a littler one there in the middle of the screen, just exited.
I have a question from Calgary, Alberta, Canada, and they're asking, do the deep sea samples survive the ascent to the surface?
I'll let the Nautilus crew answer that one since they get to see how it arrives
Get
on deck.
pretty far out in front.
Yeah.
The answer is sometimes. Usually they do. Depends on how big they are. Depends on what type of animal they are.
Do we
Pressure is
wanna
not really the
move or
concern.
keep searching in this area.
I'm in the science chat.
Yay, Julie!
I think
I'm
the
in.
I think the goal was to get a 3rd snail from this
Far
area.
left.
Oh.
Far left.
Yep. Oh,
Yeah.
there we go.
So the pressure is not really the concern. It's more of a temperature issue. And our boxes seal pretty well. So that keeps the bottom water in the box and keeps the box at temperature as we come up through the water column. So for the most part, yes.
Great, thank you so much. We're getting some thank yous online saying, wow, you really are live. You answered my question. Somebody said there's been a lot of little black jellyfish, and have you identified those? Do you have their names?
Yeah, there's a couple of different species here. One of the smaller ones that are very close to the seafloor are called benthocodon. They're usually a mid-water species, but you can find them close to the seafloor as well. But there's also some large red kind of globular-shaped jellies, and those are Paralia, different genus.
All right, let's come wide.
long reach.
Do you want to zoom on it before you grab it?
Do I need to come in a little bit, or do we have enough?
I think we got enough.
Covered in hydroids.
This looks like a different species.
It does.
Is that okay?
That looks more whelk-like.
That is more whelk-like. I'm waiting to see if Daniel responds to my message in the chat.
And I see that message, Lizzie.
Aw. Welcome to the chat, Julie.
Roger that.
I feel like I'm part of the team now.
Yeah, he said he'll take it.
All right.
It
Do
is a
we-
whelk.
Mm-hmm.
Steve, are we doing this as a different sample number since we're...
We're going to stay on the same sample number.
Okay, it's okay that it's farther
Yeah,
away.
same sample, and then we can move on. This is wonderful. Thank you all so much for your snail hunting skills.
Uh-oh.
That one's bigger.
Might need to rotate it, put it down.
Want me to turn off
slurp it up
suction?
the
Yeah.
longitudinally.
Let me
Hot
get
dog
it.
style.
There we go.
Oh, is it a snail? It's not a crab, right? No, because we saw the snail track. Okay.
That's the operculum.
Yep, same jar, same sample number. A-okay.
That's
Uh-oh.
pretty big. Do we dare? We can put it in a
Or
box.
we can put
Oh,
it in a box.
yeah.
Yeah, we don't want to clog
Yeah.
the slurp.
What box is it going in?
Let's come wide out.
You can throw it in any box in the forward.
Okay, let me move the camera back. There's nothing in it, right?
Nothing in it.
Cool.
Mm-mm.
Tool tray going out.
Suction off.
Suction off.
Nicely done.
Alright, tool tray coming in.
Did that one go in the left or right? I didn't see.
I think left.
Left.
Looked like left.
Wonderful.
Okay. So now we're going to zip to BG, correct?
Yes, please.
So
Alright.
Daniel's excited about collecting the whelk because they're really trying to sort out different taxonomic, the different taxonomies. So fresh DNA material is really welcome for what they're trying to do.
Welcome.
So that's great.
Well-
Welcome.
That's what I said when she joined
Oh,
science
sorry.
chat.
Oh, sorry, Paul. You get original credit. I didn't hear you. I did see you look at me, though. And I was
Yeah.
like, I don't know why you're looking at me like that.
I was like, okay, I guess my joke missed with Sam on this one. 3 minutes later, you're stealing it.
Original joke credit
She
goes
just
to Paul
pretended
Glick.
to be unimpressed.
Yeah. Alrighty, ready for a
Maybe I'll-
move?
Yep.
We're going to do a bigger step
well-
since we're going to zip in.
Yeah, what's the move?
We're going to do 046, please.
Okay.
Bridge nav.
It's like we gotta wait up.
Yeah, but I've got to...
50 meters, bearing 046.
We've got a lot of wonderful questions coming in here, and this is actually one I was wondering about when I was on watch. We have a very enthusiastic explorer who said she's been following along for the last couple of days and says, when you take the water samples, you mentioned that the water is always in there, and then you close off one to capture the water in that area. Why is it that way around rather than being empty and then opening it to capture it?
Well, so in large part, we really don't want any like empty air spaces in the ROV, because when you then descend down to these crazy deep depths, there's just this insane crushing pressure. So it's much easier to have everything be filled with water and then capture the water that you want. We can
Thank
also
you.
put up pictures of the Niskin or the video feed from the Niskin rail, I think.
there has been some samples done with pressure bottles that are you open a valve and let the seawater flow into it.
The Niskin bottles are on channel 3 right now.
Yeah, but in order to do that, you got to have a really strong, like titanium or stainless steel bottle that, with like heavy duty valves. And
Yeah, that's interesting. You mentioned the pressure increase in the hydrostatic pressure, because I know in past cruises we love to send down styrofoam cups and objects to these deeper depths to see what the sort of forces of that pressure are. So imagine that on a styrofoam cup, but if you have a bottle that's filled with air too, that could be detrimental to that bottle.
Yeah, and with
That
the
totally makes sense.
We have that snakehead eel pout.
there's also a thing called sympathetic implosion, where if you have something that, like a bottle like that, and it implodes from the pressure, it'll cause a differential pressure on other things near it, and you could get like a chain reaction.
Ugh.
Yeah,
wow.
That could be catastrophic then.
that's a big deal when you're in a submarine.
Yes.
Sympathetic
So from here you
implosion.
can
Yeah.
Definitely a band name.
I've
Yeah.
never heard of that.
Yeah, that's my new term for the day.
I was gonna say band name for 2020.
sympathetic implosion. That does sound like 2020 in general.
Yeah.
Yeah,
Oh, let's come up a bit, Bob.
I just wrote it down on my pink sticky note. I'm going to use that in my vocabulary a few
Still
times.
pretty far out.
Yeah.
Looks like another eel pout. That little silverfish that swam across.
Here's a question maybe for Julie or for Sam, from a college student saying, do you ever do internships for college students? Or if you used to and can't now because of COVID, would you start doing them again later?
I'll take the Nautilus part of that question and throw it over to Julie for other programs with sanctuaries.
That sounds great. You're on, Sam.
Thanks. Yeah, Nautilus and Ocean Exploration Trust, which is the nonprofit that owns and operates Nautilus, does have a science and engineering internship program for community college, undergraduate, and graduate students in several different tracks, including seafloor mapping, ocean science, ROV engineering, video engineering. And unfortunately, internships were deferred for 2020 to 2021, which means that we're hoping to be able to offer those spots to students next year, which means that we didn't open the application season for 2021. So do stay tuned. We're hoping to reopen, as like everyone adapting to life with COVID-19. Hopefully we'll be reopening that program in the future. And I'd also encourage folks interested in that internship program to check it out on our website just to get an idea of what kind of opportunities the internship program offers, but also to look at our live event series that we've had over the last couple of months under the education tab on the Nautilus Live website. We have a whole series of events to meet more of our team and get more in depth with the career paths that some of our team members have taken. So there's been some really great live events that are recorded now that you can review and get to know a little bit more about the different opportunities that and different tracks that you can take to get involved in ocean exploration. I've got a couple more left of this year, or this season as well, before the season wraps up in December. I'll throw it over to Julie.
Thanks, Sam. Yeah, through NOAA, we have a couple different internship opportunities. Right now, we're getting ready to select a Hollings Scholar intern. And with COVID, what we've done with our internships this year is they're remote. So we're looking at, we need help doing some species list cataloging for one of our online interactive databases, Simon. And we're also looking at developing some curriculum to go along with a new interactive condition report graphics, which you can actually look at data and interpret it. And so that's through that particular program. We also host a coast intern that's run through the Cal State universities, and that's an 11-week internship that happens in the summertime, and it's a paid internship. And we usually have our coast interns focus on a specific project deliverable. This last year, our intern developed a story map for deep sea coral community habitats, and also helped us with some of our outreach with Get Into Your Sanctuary. We work with Sea Grant, California Sea Grant, and we host Sea Grant Fellows at our site as well. And those are usually graduates that sign up for those internships, and those are really about mentoring and looking at getting specific skills or experiences that'll help them in their career pathways. And so for our current Sea Grant Fellows, they're helping us with our management plan review and also with developing a citizen science project around white abalone. So there's definitely opportunities that we support within national marine sanctuaries. We also have the Nancy Foster Scholarship Program too. So check our website, channelislands.noaa.gov, or you can also check the Office of National Marine Sanctuaries website as well for those opportunities. And so yes, in the COVID world, they've been virtual, but we're hoping to get back to where we can host interns and fellows at our site again, because I think what we try to do is immerse them in what we do as an ocean management agency and protecting the resources and engaging our communities.
Awesome. Thanks so much, Julie.
Certainly. It's one of my favorite things to do. Mentor.
There are a couple questions here about some data loggers and searching for instruments that were placed down last year. And there are 3 or 4 questions related to that. Data loggers that asking if they have been recovered or not recovered. A question for Bob, I think.
Nose goes. The question came in about the instruments that were left at Davidson Seamount that we were not able to recover. So do you want to talk about that one, or?
About why they didn't get recovered, or?
Well, the question was if they've been recovered, which they have not.
They have not.
And the reason for that is that we were, as apparently some of our long-term viewers had noticed, we were in Monterey Bay National Marine Sanctuary at Davidson Seamount. And we're returning to collect some instruments that had been left last year during our expedition near the Octopus Garden. So collecting environmental data information. I think it was temperature, salinity, probably. I'm not sure which. Maybe oxygen.
Oxygen optodes.
Yeah.
Yeah.
That had been left there to collect data over the last year, and we were just about to grab one of them and had a bit of an issue with Argus.
Simultaneous weather issue and engineering issue.
Yes, so we had to recover the vehicles immediately, but we were at 3,000 meters depth, so it took immediate was about 2.5 hours to get back up to the surface. And we were weathered out and actually came down to the Channel Islands and started dives here. And then the lead scientist for that expedition, Chad King, had decided that if we did get the chance to dive in Monterey Bay or at Davidson Seamount again, that he thought the time would be better spent doing some more of that basic exploration. And he found another organization in Bari to work with to go back and recover those sensor instruments at a different time. So we did do one final dive in Davidson Seamount on the southeastern flank of Davidson, which was had been targeted as potentially some really interesting features. I don't think it was quite as interesting as everyone was hoping for, but we did get one final dive there before the expedition wrapped up last week. And then we started here in the Channel Islands again. So a bit of a bounce back and forth between Davidson, but.
All right.
Thanks so much. It sounds like these people have been following it for a while. Several different people were asking about that. So thanks so much for the explanation.
Yeah, it was very, very dramatic. It was like the arm was stretched out to grab the sensor. We had picked it up and then had realized that we didn't have time to to do anything else before getting back to the surface, so I had to set it back down.
We're not talking about why?
Oh, you can talk about why. Yeah.
Oh,
If you want to go into detail.
okay. I didn't know. Yeah.
Yeah.
So what happened was we saw a leak indication on one of Argus's main electrical housings. And that's a bad thing. Leaks are bad. So, so we also saw the humidity sensor in that electronics bottle go to
120 or something.
120% humidity, which is also bad because it normally runs around like 10 to 20% humidity, very dry in there. And seawater and electricity don't really, well, I guess they get along really well. I don't know.
Too well.
Yeah. So that was a bad thing. So that bottle ended up being completely flooded with seawater and wiped out all the electronics in that bottle. And we had to do a huge effort to rebuild all the electronics in that bottle in 3 days. And here we are back in business. So
Yeah, it was incredible. The whole ROV team of 6 people were basically working night and day to get that back online.
And we were lucky to have most of the parts for it, but had to make a detour into Catalina to pick up some parts that we had FedExed out and had somebody run out on the fast ferry.
Yeah.
And yeah, but I think we did pretty good there.
It was an impressive on-ship and off-ship logistics effort.
Wow, that's amazing. I want to let you guys get your work done, but there's so many questions coming in. So maybe, why don't you just tell me when you're ready for the next question, and then so I'm not taking over your entire time.
Well, I think we're kind of just cruising along to the next waypoint. So science, I mean, definitely interrupt if you see anything, but from an operations, we're just good continuing. So.
How long has it been since our last transect?
Let's see. We have gone 150 meters.
Okay, why don't we set up to do another, and then those are usually pretty quiet since we're just fixed heading and fixed speed, so we can talk after we set up another transect.
Sounds good.
Same heading, 046?
046. Okay. Looks like it'll be bearing and heading.
Yeah, for a transect. Yeah, it's nice when they're the same. I'm ready anytime.
Okay. How about now?
Good.
Transect starting now. Transect 2B.
Thank you.
Okay, I have a question for Lizzie. Someone was watching you do that photo mosaic that you did around that really beautiful Lopelia reef yesterday. I think that was yesterday.
Oh, yeah.
And asking if there are any examples of what a photo mosaic might look like. Is there one that people can find now online?
There's several. So let me make sure I'm pointing people in the right direction. So originally, the first one that I encountered that was created by sanctuaries through Nautilus footage was the Davison Seamount whale fall from 2018, I think, or 2019. 2018.
2019 for the whale fall, 2018
19.
for the octopus garden.
Got it. Okay. So 2019 for the whale fall. And Chad King, who was the PI of that cruise, has been working with another institution, the Monterey Bay Aquarium Research Institute, or MBARI for short. And the MBARI contacts, oh shoot, I think his name is Ben, had helped take the YouTube video that OET uploads to their YouTube channel and created his own screen grabs or frames from the video, and then he used a software to overlap them essentially, and create this whale fall mosaic. And it's all publicly available. And MBARI does this with their own vessels and ROVs as well. And so if you go to sketchfab.com, it's a public webpage, and all kinds of institutions and researchers and anyone else who has access to the software kind of hosts and uploads their photomosaics. So if you go to sketchfab.com and you just type in whale fall, you'll see the rosebud whale fall and the whale fall at the Davidson Octopus Gardens, which both of these are done by MBARI. And then if you click on MBARI, once you've gone to one of their mosaics, you can kind of see more that they've done. And they've not, in addition to this whale fall, they've also looked at, they've made photo mosaics similar to the one that we were trying to do last night, which is like capture images as you circumnavigate a boulder with interesting corals and sponges on them. So if you go to sketchfab.com and you either look up MBARI or you look up the whale fall, there'll be one of the first two images that shows up in your search.
And the whale fall mosaic is also linked on the Nautilus Live website under education in our education resources.
Oh, perfect. That's even better.
Great, thank you. Thank you so much. There's a question here about from an aquarium volunteer who's asking if there are any opportunities to participate in crowd research. An example like cataloging fish from scans or transect data or something like that.
I'll speak for Nautilus. We are the data collectors. So we provide all of our data to all the science teams that we work with for their use. And we do know that there's a lot of work that goes on months and years after that data is collected with video review and sample processing and continued study. And I know that there's been a handful of times in which there has been specific projects that scientists have kind of crowdsourced out to complete that data processing, but we don't have anything, we don't have any mechanism for, at this point, having our data crowdsourced. There is a project called Ocean Video Lab that's in kind of a beta test mode, and some of our footage is in that, along with other exploration ships, including Okeanos Explorer and Falcor's, or Schmidt Ocean Institute's Falcor. So I'd recommend checking out Ocean Video Lab if you're interested in getting involved there. It is a beta test project, so I'm not sure where it is at this point. I'm not sure, sanctuaries, are there any opportunities to get involved with crowdsourced projects on your end?
For sanctuaries, not with fish so much, except for, well, I take that back. There is the Spotting Giant Sea Bass project. That's for divers to go out and actually, they're trying to, capture images of individual sea bass. They're able to look at unique identifying features on individual giant sea bass and be able to track them over time where they go. We know they utilize the Channel Islands and go between islands. So there is that sort of citizen monitoring effort. And I know with the whales, with the whale alert app, you can go out whale watching and you can, if you see a whale, you identify it. You can go ahead and enter in the information, including the latitude and longitude, and that's helping us sort of understand the distribution of different whale species within the sanctuary and the surrounding Santa Barbara Channel at different times of the year. And then I know, iNaturalist is a big one for looking at intertidal organisms. That's not necessarily sponsored by NOAA, but we have our limpets monitoring, which we were talking about earlier, where we work with youth to go out and collect intertidal data for both the rocky intertidal and sandy beach. And so there's a few opportunities within NOAA.
Yeah, and as far as being able to do some of this stuff from home, I know we're kind of just dabbling in the machine learning. So looking at images and having people from the public point out that's a rockfish, that's a coral, like very generally, so that we can train machine learning algorithms. But I think kind of like what Julie was getting at, there's more opportunities like that. Tom Leidig with the Southwest Fisheries Science Center just pointed out there's also something called REEF, or Reef Environmental Education Foundation, which is also for scuba divers.
Yes, and
They're
they've been
counting
helping
and
to
identifying
do the
fish.
fish surveys inside MPAs, marine protected areas.
Oh, Daniel just put in a science chat one that you can do from home, which is nice to add to the repertoire that we're talking about here. It's called Notes from Nature, and you transcribe a collection of data from museums. So you can be helping out different museums from home, which could be a very nice quarantine activity. Still feeling productive and something you don't have to leave your house for if you're not comfortable with it.
That's super interesting.
Those
Notes from nature.
fantastic.
Writing that one down.
Thanks,
Yeah.
Daniel.
So I have questions coming in. The interesting thing is, Sam, I'm going to the website and trying to refresh, and I'm actually getting some messages in here saying, where do I go to ask questions? It says that the team's not available, but some people are asking questions anyway. So I've got everything updated and available, like from my side of the website, but maybe something to look in for the next Q&A.
Yeah, I'm
section.
actually on the website right now and it looks like it's green. So I'd encourage folks to refresh their browser, and it could just be a caching problem on individual computers. Hopefully that'll help.
Fantastic. Thank you so much.
Yep.
There's quite a few questions on if you've discovered any new species on your explorations and how often that happens.
From the sanctuary perspective, while we're on Nautilus and doing this kind of work, it's not super often, but we did discover a new species of coral last year. While a lot of researchers were in Cordell Bank, it's also been found in Channel Islands National Marine Sanctuary. And we also discovered the purple orb outside of the sanctuary in Arguello Canyon a few years ago on the Nautilus. So once every few years, but we're not out here as often as the Nautilus is. So I'll turn it over to you guys if you have an answer for that.
Yeah, I can answer a little bit more. The answer has been yes, that new species have been found, and typically this is not a fast process from the point at which we get it up onto the ship to the point at which it becomes described as a new species is often years to sometimes longer. But we have initial data usually when we're on the seafloor that something looks odd or strange, or it's not quite what we expect for that area or for that particular taxonomic group. So we have some initial data that suggests that it might be a new species, and then that can be followed up by specialists when it gets to the repositories.
Great example.
Yeah, I mean, for example, the mysterious purple orb, which is probably a type of a velutinid snail that we collected in 2016. It's at the Harvard Museum of
Comparative Zoology.
Comparative Zoology, and it's still being described. But they do think it's a new species, but we don't know yet.
Yeah, and then there was, in 2015, Nautilus was in the Galapagos Islands working with researchers from the Charles Darwin Foundation. And just this year, a paper was published identifying and describing 30 new species from those expeditions. So definitely takes, as you said, years to find out what exactly you have.
Yeah, that actually, I think that paper came out when I was in quarantine. It was pretty recent.
Yep, yep.
And they identified 30-something new species, but they're not all described yet. So
okay.